Oh no. Hey. Come on in. Hey, Spray Nerd Kruger. Welcome. Hey, everybody. I am John Beckmann, Professor John Beckmann, Dr. John Beckmann, U.S. Senate GOP primary candidate in the state of Alabama. Vote for me March 3rd. Today, we're going to talk about genome editing human babies some more. I talked about this before in one of my last videos on genome editing dogs. And we'll talk about it a little bit more. But first, we'll talk about the issues, the problems, what concerns people. But first, if you got something to drink, Sunday night, cheers. Let's have a citation to the simultaneous libation of Scott Adams. Oh, so good. Okay, so in the last videos, I talked about how the Russians and the Chinese are now openly saying they are editing human babies. The Chinese already have, and the Russians are saying, well, if the Chinese are doing it, we're going to do it. So there's two governments that are essentially saying, like, we're going to be editing human people. And so my last video talked about what should the U.S. be doing? Like, knowing that other governments are doing this, what should we be doing as leaders in research, as leaders in science? What should the U.S. be doing? Should we be doing human genetic editing of people? And I talked about how the Chinese, the reason that the Chinese did it is they had families that had HIV/AIDS. So the father had AIDS, and the father wanted to make a child the natural way, but then that child has a risk of inheriting AIDS. So what they did is they took sperm from the father, an egg from the mothers, and they did in vitro fertilization in a test tube. And then they micro-injected the mechanisms of editing, so this is called CRISPR-Cas9. They micro-injected the Cas9 enzyme into the human embryo, and then targeted a very, very specific gene called CCR5, which once they mutated, gave that embryo resistance to the HIV virus. So what they essentially did is the Chinese essentially engineered human embryos that are resistant to the HIV virus and resistant to AIDS. Okay, and then now the Russians are saying we're going to do that too. So I just this weekend read this other article. This was also in Nature magazine. This is more talking about more applications of this system and other issues with the system. So this is also applicable to Huntington's disease. So Huntington's disease is a disease like Parkinson's, where if you have it, usually you have only one mutation in a single gene will give you Huntington's disease. So that's what's called dominant. If you have just one mutation in that Huntington gene, you'll get Huntington's disease. And it basically, your neural systems in your brain start declining once you hit the age 30, and you basically just kind of progressively become more and more like a vegetable. That's Huntington's disease. So people have proposed that we can use this CRISPR-Cas9 system to essentially get rid of Huntington's disease. We can essentially engineer all embryos to not have Huntington's disease, knowing whether or not if the father was a carrier of the Huntington mutation. Okay, so this is a good application of it, but the question is, like, why not? Like, why would we not do this? Why are essentially we not doing it right now? So the first point is that this is embryonic medicine. So it's almost like we've never had this type of style of medicine before, where you take an embryo, you find out if the embryo has a particular disease, and then you fix that embryo, and then you implant it, okay? So that's embryonic medicine. We've never really had the technology to do that before until now. So it's brand new. The other thing is, so when you inject the Cas9 molecule, what this is it's a nuclease. It means it cuts DNA. okay? And we've controlled it in special ways where we can tell it where to cut. But that specificity only goes so far. So when they take this embryo and they microinject it with the Cas9 editing enzyme, there's an off chance that it can cut the wrong piece of your DNA. So it can give you mutations in other parts of your genome which you were not planning for. So the efficiency and the specificity of the treatment, you could call it, is not 100%. Those are called off targets. Now, one of the other problems is called on target. The other problem with the system is we can tell it what to target. But then once it hits that particular gene that we're trying to fix, that we're trying to repair, we can't tell it precisely what to create. Okay? We can't tell it what to insert with precision. We're working on that. We're getting better at that. But as of now, we can kind of just tell it to hit this piece right here, which is broken, and we have just a chance of fixing it. So there isn't a 100% chance of fixing it in the correct way. That's an on target. So there's on target unpredictability. Okay, now all the research in the system is working out these kinks and optimizing the system in mice, okay? So mice are a model system for humans because we can do all these experiments that we can ethically do with mice that you would say would be unethical with a human being. So for instance, nobody really cares if you're genome engineering mice embryos, but they would care if you were genome engineering human embryos. So we spend a lot of time and a lot of research money and a lot of investment optimizing these systems in mice. But mice are not humans. So there's all kinds of treatments that have been developed in mice that never make it to humans because it might work in the mouse, but then when you put it into a human is not a mouse, and so it maybe no longer works. So we're spending all this time optimizing in mice, and optimizing in mice is not necessarily going to be optimized in a human embryo. So that's one of the problems is there's no way to just like figure it out beforehand. The only way to do this is with human embryonic experiments, which is why this makes people very, very, very nervous, because now you are experimenting with somebody's life. You're experimenting with a human embryo. So this makes people very, very nervous, understandably, because if there's a chance to cure a problem, but there's also a chance to create a new problem. You could create an entirely new mutation, and now you have a child that has some kind of new mutation that you were not expecting. But the only way to figure it out is to do the experiments in the humans. So that's the catch-22. So here's what I was thinking. It's very, very complicated moral discussion, because we could use the technology to literally eliminate completely from the earth certain genetic disorders like Huntington's disease. We could get rid of that. It would no longer be on the planet unless it spontaneously arose again later. Okay, so we could use it for that. And the cost of that, the cost of achieving that advance to human health, would be the sacrifice of many, many, many embryos, okay? So that would be the cost. And I'm not saying that that is ethical or not ethical. I don't know. I'm saying imagine a future where there was no longer Huntington's disease. We could make that. But the cost of that would be the sacrifice of essentially human embryos to figure out how to make it work. That's what would happen. So I'm just talking about it now. So here's the funny thing is, this is why this is kind of like this application that I'm talking about now with Huntington's disease and these HIV things. It's kind of like pseudoscience, or not pseudoscience. It's kind of like pseudo treatment. It's like pseudo medicine, because the alternative for the parents who had the HIV, or the alternative for the parents who had the Huntington's disease, is that they could have just got a vasectomy and chosen not to pass along their deficient genes or their HIV virus, okay? So the alternative, the safe thing that wouldn't cost any human capital in the human embryos is these people just get vasectomies and they don't produce offspring, okay? So sterilization. That is a way that you could cure these same diseases without crossing that ethical boundary. So again, it's kind of like a pseudo treatment in a sense that like there's other better ways to sort of deal with this. But people are using these experiments as an excuse to test human genome editing technologies. Which is why I said in the last video, I don't think I'm at the point yet where I'm ready to say we should be doing this in humans. I'm not there yet. So I'm going to say we should not be doing it in humans, but I did say we should be doing it in dogs, and I gave specific examples of that. We should be doing it in animals. We should be doing it in mice. We should be doing it in our agricultural systems. We can do this in our agricultural cows or chickens to make better agricultural products. And we can use these as systems to develop the technology, but we're not ready yet to use it in humans. It's going to happen eventually. There will be, I should say, it already did happen. In China, they already did do it. In China, they already did make the human genetically modifications, and they're only going to increase the technology more. They're only going to use it more and more and more and more. So you're only going to see this more in the news. You're not going to see it less. So I'm just telling you about some of the costs and some of the things about it. So I'll end there. Also in Japan. You got any questions? Hi. Welcome from Japan. Let's have a cheers. Sip. We're talking about human genome engineering, editing human embryos. Any questions? Otherwise, I'm almost done with my cigar. I can finish. Tomorrow, I'll talk about the shootings that have happened. I'm not going to talk about that today because it's a bad topic. I'm not going to talk about it today. So what are the real benefits of that today? The benefits of human genome engineering is we could get rid of certain genetic disorders from the population completely. So we could literally feasibly eliminate Huntington's disease. We can create humans that are resistant to the HIV virus. I already talked about that. Those are some of the questions. Hello, Russian. What's going on? I'm learning English listening to you. Do you have any questions about America? Well, you're learning from a bona fide professor. I'm a professor at a university here. What's your topic of study? You're learning English, but for what purpose? That's it. I said my thing. How do you make sure corporations don't manipulate the genome with artificial intelligence? Why would we want to stop corporations from manipulating the genome? I think we don't want corporations to manipulate human genomes, but we definitely want corporations like Monsanto to manipulate agricultural products. So we definitely want like agricultural companies using machine learning, using genome editing to make better plants that we grow, to make better agricultural products, to make better pigs, to make better cows. We do want that. So that's not something that we don't want. That's something that we do want. But we do want to not allow them to edit humans for sure. Any other questions? That way we can select the sort of people we want. Smart, good. Well, that's what they're... I tweeted that. This guy's funny. So I actually tweeted that this morning and I deleted it. I said, there's actually been a study that says your political affiliation is partly genetic. So by sequencing somebody's genome, like the 23andMe stuff, they can make a very, very accurate, pretty close prediction of what party you essentially will associate with. And so I tweeted that. I said we could use genome engineering to essentially almost ensure that all of our children would be Republican and or Democrat if we wanted to do that. Although that would be a pretty bad idea. But we could do that. Didn't see what that other one was. It's not letting me go through the other comments. Any other questions? Otherwise, I'll end it. My cigar's done. Have a good night. Oh, wait. They put people in MRI and found that they can tell